I'm Alicia, the product manager of BMKGENE and responsible for the genomics product. Today I'm very glad to be here to talk about some information about the de novo genome assembly. Since de novo genome assembly is a very complex project, and so if you want to do if you want to start the assembly projects, then before the project you need some information for the preparation. So here I will share some information about the strategy formulation and also the sample preparations for you to help you. I hope it can help you to find some ideas to prepare for the project. Okay. In today's webinar, I will first give a brief introduction of the de novo genome assembly, and then you will show some indicators to evaluate how assembly result is good or not, and then we will do the part of the challenges in the genome assembly, and corresponding to this, I will share some information like how the technique has been developed to fulfill the requirements of the challenges in de novo genome assembly. After that, I will show you some assembly strategies for different level of assembly, and then after that, I will share the sample preparation and the assembly experience in BMKGENE. So that's the content today. So first, let's go on the assembly overview. So we know that the DNA is the genetic information within our body or within organism. So and the DNA is in the chromosome a state with a specific structure. So if we want to study the genome and know the information of the genome, we need to get the genome sequence like the base order of A, T, G, C. Then if we want to have an overview of this genome, then we need to get all the sequence of each chromosomes. So I would say that de novo genome assembly is a process that to reconstruct the genome sequence of an organism and from the sequencing data. So for the whole project, it should be from sample to genome sequence. So first, when you start the project, you will need to prepare the DNA extraction from the selected materials. Then you do the preparation for the sequencing. It's more about the library preparation part, since the gDNA is very long. So in the long library preparation part, we will do the fragmentation, then to add some adapters onto the fragment to construct a library for the sequencing. And after the sequencing, you will get the read sequence of each fragments. Then you can using this sequencing the reads to do the assembly and to have an overview of how the genome sequence are. So this part is mainly done by the bioinformatics analysis. So after that, we could get the chromosome level genome sequence. Then that's the overview of how it works. So next part, I would say that there is a very great leap of assembly numbers since the previous decades. You can see this is overview paper of the published plant genomes from the 2000 to 2023. So you can see that here from 2000 to 2020, there has the decrease of the assembled genome numbers, and after the 2019, there will be there was a very big increase. Here, I would say it could because of the new technologies of sequencing, like the long-read sequencing. I will talk details about this in later. And you can see that along the sequence assembly numbers, you can see the chromosome level genome assembly is very high in the last 3 years than before. That means that the assembly quality also gets increased. So for each species, you can there is the statistics of different plant class. You can see that within each class, there has some species being assembled into a reference genome in the previous years. So you can see that the assembly of a reference genome is very important in the study research. Why it's very important? From here, I'd like to say that the genome sequence could provide the genomic resource for the functional genomics, multi-omics, population genetics, evolution, breeding, disease mechanism research, and so on. So, this is for the research level, and for the genome, how to use the genome level, I would say that could be divided into 3 parts. The first one is about to assemble genome and use this genome as a reference genome. From here, a reference genome can work like a framework or a coordinate of the reference genome. And then if you want to study another individuals of the species, then can do just the sequence the genome of the individuals, then apply the sequencing rates onto the reference genome. Then you can compare the variations between the sequenced individual and the reference genome. Then you got the variation markers of these individuals. Then when you do population genetics, or you want to compare the individual to individuals, then you can align all the sequencing rates from each individuals onto the reference genome. Then you get the variations within each individual. Then use the reference genome as a standard to compare the difference or variations between different individuals as the population genetic level. So that is the reference genome part. And besides this, you go for the downstream analysis, like you want to validify the function of a gene. You need to get the gene. This usually to be conducted by some analysis like GWAS and genetic map, like that, to find the trait-associated genes. So in these levels, the reference genome is very important. You can get the gene ID and the gene positions and the gene sequence from the reference genome. If without this, you can't relate your results to the gene levels. And also, if you want to do the gene validation to test if it really has the function on the trait, then you need to get the sequence to do some downstream molecular experiments. And this time, the reference genome also be very important to give the gene sequence for the research. This is the within species level. Another within species level is about the pangenome analysis. Pangenome analysis is analysis to try to get the most represent individuals of the species. Then we assemble all of the genomes of these selected materials. Then we can get a more comprehensive genetic information of the species, since we know the individuals within the same species will have different genetic information, such as some materials is very disease resistant, but others are not. So there should be some genetic information difference. So from pangenome, we can get an overview or a comprehensive understand of how the genetic diversity within a species. Another one is about the comparative genomics. So this is studies between among the species. For some studies, like you want to compare how this species evolved and then generated to another species, and you want to have a look at how this evolution in the genomics levels. So this comparative genomics will do this, but this is also based on the assembled genome of different species. So this is the application part. So next question is about when is the genome assembly is needed. So first case is that if there has no genome has been published in this species, so the reference genome to assemble a reference genome is very important thing. After you got the reference genome, all the downstream analysis from your group or other continued will use this reference genome to do downstream analysis. But if your studied species has been published with a reference genome, you can also have the chance to assemble into another genome. Here are three cases. One case is that one commonly used individuals or variety has been published and use this as a reference genome, but you have some other materials or like other breed with very investigable traits or has some interesting biology stories need to be talked, then you can assemble different variety within the species and then compare to the reference genome to talk about how the genetic information is different from this one to a reference genome, then connect the story to how they affect the different phenotype of these two varieties. Another case, next case is that if there has a reference genome, but it's not very high quality, like it's a draft genome, it's just a kinetic or scaffold level, or it's just assembled using the old version sequencing technology, like it's assembled from the short read sequencing technologies, then you can update the draft genome to chromosome levels. That means we could get the near to the chromosome sequence, then it's a more complete and comprehensive information could be given to the species. Another case is this species already has the chromosome level genome, but the genome quality, I mean, the genome quality is just the haploid level. That means that just one set of the genome has been assembled. In these levels, if there if one species it has very high heterozygosity ratio, then where there will be many parts regions that has a different phenotype within the different chromosomes. So different chromosomes. So there will be get fused when you focused on this part and reference this sequence to do the downstream analysis. So in this case, it could be useful if we could divide this to two parts of the heterozygosity regions, then and we assembled into a haplotype resolved genome, then it's more clear or more correct to show the information of the genome and facilitate in downstream analysis. So in these levels, you can also update the assembly into a higher level. A higher level also means this is for the high heterozygosity species. And if your species has a reference genome, it's just not T2T level, then we can choose the T2T level genome assembly as the option to your project. The T2T level is just a more complete assemble level that you can, the genome has been assembled from the ceremony to the ceremony. So this is very complete. Like for example, the human genome. Previous we use a reference genome that is not T2T level. But after the work for all the institute and researchers for this research, now a T2T level genome of human has been published and show us more dark regions of the reference genome that which the information has been masked before. So this is the situation about when is genome assembly needed. So since we have talked about the genome assembly quality, so next part we will talk about how to evaluate assembly, it's good or not. So there has some indicators to describe the quality of the assemblies. The most important three parts are like the continuity, the completeness, and the correctness. For the continuity part, the more used one is the N50 value. The N50 value is the length for which contigs no shorter than this number cover half of the assemblies. You can see here that after the assembly, we ordered the contigs from the longer to shorter ordered by the length. Then we add the contiguous from high to low. Then when we add this one, add this one, add this one, when the total length can reach the half of the total genome size, then the length of this contig here is the 60 will be the N50 value. It's an indicator to show how the continuity of the assembled genome. If it's a perfect finish or say finished genome assembly, you can see just one contig, and with the one contig can correspond to one chromosomes. Then the N50 could be equal to the chromosome N50s. Also, if one chromosome is constructed by very less contigs numbers, so the gap numbers will be very low. If the gap numbers is low, it shows that the assembly is very high quality. So here I mentioned here that if it's a very good assembly, the gaps could be zero. And now the zero gap reference genome is the purpose of many genome assemblers. And also here has some new indicators for the continuity evaluation, like the CC ratio is about the contig number compared to the chromosome numbers to show the continuity. Yes, next part is about the completeness. The completeness we can evaluate in two ways. The one is about the overall completeness. The first one is about the genome size to see whether the assembled genome is very related to the real genome size. Another one is the chimer-based method. You can see here, after assembly, then we can do the chimer analysis in both the assembly results and the sequencing rates. We will count the chimer that found only in race and the chimer found in both assembly and race. And then we use these ratios as indicators to show how the quality of the assemblage if you can imagine that if all the sequence has been assembled into the genome, then the ratio will be 100%. It's a very perfect value. So if the value is near to 100%, we will see that it's very good assembly results. Next is about the gene space completeness. It's not in the sequence level. It's about the gene that has been sequenced or predicted in the reference genome or the assembled genome. You can see here we use a Busco database to do the evaluation. So it works by aligning the conserved single copy proteins to the genome and containing alignments that are missing, broken, or duplicated. You can see if the assembly is very completeness, that the conserved genes or proteins will be founded or be aligned in the reference genome. You can see if it's a very good or perfect assembly, the values could be near 100%. But in real cases, if the value is more than 55%, we would say that it's a very, very good assembly. Next part is about the correctness. There are also two commonly used values. The first one is about the single-base quality value. We called it QV to see how the sequencing rates quality and the correct base ratios. Another indicators in this part is about the sequence rates map-back ratio. When we do the sequencing rates map-back, if the assembly is very correct, then all the sequencing rates will be mapped into the assembled genome. If the ratio is about 100%, wow, that's a very good assembly. But for most projects, we would say that it's more than, also we would say it's more than 95%. We would say it's a very good assembly. With the assembly methods being developed, so there are still many other indicators to describe the assembly qualities, like the original genome and the heterozygosity and the high-C genetic map, whether they show a very good assembly results, and also the reference-guided collinear rate analysis. It would say that if your assembled species has the reference genome before or have some related species has a reference genome, then we can use the assembled genome and to do the culinary analysis with the published reference genome to see if they have a very good culinary rate. If it's very high culinary rate, then we would say that the assemble is good. So this is this part. So since we know how to describe a genome, it's good or not, the next part we would talk about the challenges in genome assembly and to see how our technology is being developed to meet the requirements of the genome assembly. The challenges in genome assembly could be described in three parts. One is about the high repetitive sequence content. Next one, high heterozygosity, and the last one is a polyploidy. If a species has a 60% or more than 60% high repetitive sequence content, we would say it's a very complex genome to be assembled. You can see here, I show that if there are repetitive sequence like the fragments can take two, the blue one, when we do the assembly, since the sequencing rate is shorter than the repetitive sequence length. So when we do the assembly, the kinetic tool, we could not decide which one is being connected to this kinetic, like the blue one is next to the green one or next to the blue one. So there will cause the difficult to assemble the high repetitive sequence content genome. And another one is the high heterozygosity. If the genome regions that from the maternal one and from the paternal one, they have different nucleoside or they have different regions or fragments. Then when we do the assembly, they will also be confused to which one should be which one should be located in this position, and when we do assembly, will cause some bubbles here. So for this part, if we can see if the sequencing race is longer than the repetitive sequence region or the high heterozygosity regions, then just one sequencing race will tell us how this part looks like and how this blue one next to green or next to the blue one. It will show us the directly sequence. So if we can purchasing high or if we can using long read sequencing technologies, then most of the repetitive sequence or heterozygosity sequence shorter than the sequencing read length will be solved in this case. So for the challenges in genome assembly, we need to develop the sequencing strategies for longer read lengths. And also for the high heterozygosity, the bubble confused ones, we need to do the to develop the more efficient algorithm or softwares to solve these problems in the bioinformatics analysis part. One algorithm named the redundant is to help assemble to the haploid genome. They will just using some algorithm to move the redundant in the bubbles and just assembled into the haploid genome. But for higher purpose, if you want to maintain the different genotypes within the high heterozygosity regions, then you can using the phasing algorithm to assemble to the haplotype resolved genome. That means we will assemble into different set of the chromosomes that to maintain both the genetic informations within the bubble ones. And also, I'd say that long read sequencing has different methods for this purpose. So we need to develop the more efficient, high compatible methods or softwares to do the analysis or assembly of the long read sequencing data. So yeah, this part we need to find or use the more efficient algorithm or software for the assembly. Another technologies for sequencing developed for used for the genome assembly is the Hi-C, or one of the long-range sequencing technologies. It can sequence it can capture the interaction part of two long-range regions. Then they will be captured and fixed in the tissue state. Then we can do the experiment, then just like a cut, then the purification like that to just maintain a sequence that have half past the regions in this site and half past the regions in the interact site. Then from the sequence we could know the spatial actions between the long-range sequence. For these technologies, we could know that which regions or kinetics are within same chromosomes and others are not, and we can do the alteration and orientation and order of these kinetics to assemble chromosome level genome. Also, these technologies could give us the phasing informations when we do the haplotype resolved genome assembly. Yeah, and also for polyploid species, if it's a allopolyploid species, it could see less complex because we can get the different genotypes from the reference of the parental genome sequence. But if it's an autopolyploid species, I would say it's very complex to assemble the genomes, that there are very similar regions in different haplotypes, and then we need to divide them into different haploid type using the high performance algorithm or softwares. So it's very complex compared to others, but we also have successful cases for this part. If you have some species that is very complex and want to assemble it, then you can give us more information. Then we can give some ideas or recommendations for this part. Yeah, to summary, we could know that for the challenges in high genome assembly, the high repetitive sequence content and the high heterozygosity and the polyploidy, then we need to select the suitable sequencing technologies, and then to select the suitable algorithm or softwares to do the assembly part. So this is these two parts. Since the sequencing resilience is very important for the assembly, so here I show you how the sequencing technologies has been developed. So first one, technology or see the first generation sequencing technology is named the Sanger technologies. It has the resilience of about 600 to 1,000 base pairs. But the disadvantage of this method is that the throughput is very low for this method, even though it has a very high accuracy ratio. And works as the standard, gold standard for the sequencing. But yeah, it's very low output for this method. So after that, the technologies has been developed into high throughput sequencing. At this time, it's the short rate sequencing. The standable platform is like the sequencer from the Illumina and from the BGI. They can generate the short rate sequencing rate. It's about 100 to 150 base pairs in each rate. It's also very high accuracy, but the sequencing rate is very short, and for a higher quality of genome assembly, we need to purchase some higher sequencing methods. Then after that, the long read sequencing method, I'd say the PacBio CLR mode sequencing and the ONT common mode sequencing, it has achieved more than 10 kb or 30 kb base pairs for the sequencing rates. So for this one, it can solve the many problems that are confused by the heterozygosity or the repetitive sequence. But the disadvantage of this technology is that it has a very low accuracy So for the assembly using this method, we need also other the high accuracy rate, short rate sequencing data to correct or polish the assembled genome to higher levels. So it's very hard work to do the assemblies, but the cost is relatively lower than the hi-fi or other next generations technologies. So you can see here when it's about 2019. Here has published of the hi-fi rates sequencing of the PacBio. It's the CCS mode. It has advantages of high accuracy and the long rates. Both covered the disadvantage of the NGS and the TGS sequencing part. So you can see here is a comparison. You can see that for long rates, it's the PacBio CLR or ONT. It has a longer sequencing read length, but it's very low accuracy. Short read is high accuracy, but very short read length. So, but the hi-fi rates covers both the two advantages. And here is the summary of the plant genome assembly in the last 23 years. You can see that most of the technologies is used in the CLR mode or other ONT like this, the other TGS sequencing modes, and compared to this, the new released hi-fi rates could get a higher N50 values or say higher quality genome assembly. And this is a very new technologies that is ONT ultra long rate. The N50 value of the sequencing rates could be reached more than 100 kilobases, and the max read length could reach to more than megabases length. So it's a very useful for the assembler to overcome the heterozygosity or the repetitive sequence that is the regions is longer than the hi-fi sequencing rate length. This will help to solve the problems that hi-fi PacBio CCS could not resolve. So combine the hi-fi rates with ultra long rates. It's also another option for assemble a genome. You can see here, since it's a very new technology, so for the ONT sequencing, here the ONT sequencing refer to the ultra long rate ONT sequencing. So you can see just a few papers has been published using this method, but the quality and the N50 values is very high compared to other methods. So this is the one I said to another sequencing technology. It's about the long range sequencing technology. We can capture the interaction part between the two chromosome sites. Then we fix them, and to do the you cut this using the enzymes. Then we can get the sequence that half the site and half the interact site. Then to do the sequence from the sequencing rates, we could got which part and which part are connected in the chromosome spatial state. After that, we could give some assembly strategies for you to reference to assemble a genome. So for a common genome assembly strategy, we would say, first one, you need to do the genome survey to get the genome features of like the genome size, heterozygosity, repetitive sequence proportion to get idea of whether this species is complex or simple or like that. like that, yeah. And for this part, we recommend 50x NGS sequencing to do this service. After that, we could set the sequencing strategy. If it's very simple, then we can just recommend the 30x hi-fi data and to do the draft contig level assembly using the sequencing race. And then if you want to update the assemble into a chromosome level, then we could use the 100x high-C race to do the assembly into the chromosome level. You can see here I give a figure to show how this progress works. You can see from the genome DNA, we do the fragmentation and the library preparation and the sequencing. We got the reads sequence. Then from the reads sequence, we could using some algorithm like the overlap or some DBG method to do the assembly. If they have got connections between each others, they can be assembled into the contig level. So different relations parts will be assembled into different contigs. And when we do high-C sequencing, from the data, we could know which part and which part is interacted. So from this interaction information from the data, we could do the order and the group and the orientation of the contig. And from the contig, we could update the assemble into the chromosomes levels. At this part, we will got the genome sequence of the genome assembly. After that, we could do the genome annotation. In this part, I mean using mRNA-seq data or other reference genome annotation informations from the related species or some de novo predict de novo predict method, then we can do the genome elements predictions. One is for the coding gene part, which is very useful for downstream using, and another like the non-coding genes prediction, and also repetitive sequence like the random repeat and the transposons. These elements could also be predicted. I would say that this is a common level of genome assembly, that if it's a higher complex species, we would recommend higher depth for the assembly. And also this by default is to assemble into a haploid genome. If you have any other request, like it's a very low heterozygosity and has very low repeated sequences. We could try to assemble the genome into a T2T level genome. That means that building a zero gap genomes to obtain the most complete structure and information of the species. And also, as I mentioned before, the haplotype resolved genome assembly for the high heterozygosity species, we can distinguish the homologous chromosome pairs and performing haplotype assembly of diploid genomes. And also for the polyploid genome assemblies, we can also do for the auto1 and the L1, just assemble into the subgenome levels. Here has also a very complex example. It's about the larger size assemblies because this will use more data and has a higher requirements of the compute resource. So it's also very complex if the genome size is very high, but we have the experience on this. like the note I take here, like the low heterozygosity, low repeated sequence. This is recommendations of how the possibility of your species genome could be assembled into this level. But if it's out of this state, like the heterozygosity and the repeated sequences, it's just a middle state, then sometimes it's unable to optimize the assemble into higher level based on the current sequencing technology and assemble algorithm. So we recommend you to more focus on the downstream biology stories talkings. Yeah. And for different levels of assembly, we have the different assembly strategies. For T2T, from here you can see we will use the double coverage HiFi data to do the assembly, and also use the ultra long rates to do the gap fillings for to get T2T levels. And for the haplotype genome assembly, we have two methods. If this one, you can get the parent informations or get the parental and WGS data, we can use this method to just first using the parental data to do the splitting homologous gene sequences, and then use the Hi-C interaction signals just to assemble into a high quality haplotype genome assemblies, and then do the gene prediction and then annotations. But if you have no parental information, then we can just assemble the chromosome level genome in default. Then we use the Hi-C interaction signals to splitting the homologous gene sequences and to do the haplotype resolved genome assemblies. This is this part. And also for the polyploid genome assemblies, we also have the strategies. But for here, I would say if you want to assemble into the subgenome, then the sequencing data, sequencing depth should be the be of each subgenome size. So you see that for here just this 30x for each subgenome, then totally it's totally it's the 60x than previous default sequencing depth recommendations. Here I show some genome assembly demo result. You can see here is the most important result of the genome survey. From this figure, you could get the heterozygosity, repetitive sequence percent, and the genome size from this survey. And this is the Hi-C heat map after the assembly. You can see if it's a very good assembly, then the Hi-C signals will be very significantly higher in the diagonal lines. Because the interaction signal within chromosomes will be the most higher. And after that, the outer we will show the circle plot. The outer circle is the genome sequence of each chromosomes, and the intercycles are the predicted genes or non-coding genes or elements like the transposon or tandem repeat, their distributions on the reference genome. These are the most important result of genome assembly. So let's move to the sample preparation part. For the samples used for genome assembly, we need to prepare three parts. One is for the genome DNA. It should be a high molecular weight DNAs. This is used for the sequencing rates, for the NGS for the genome survey, and the TGS for the HiFi data sequencing. hi-fi data sequencing. And also for the tissue sample preparation is used for the Hi-C experiment because it uses the tissue sample to conduct the downstream experiments. And also for the total RNA to do the mRNA sequencing to help to do the gene predictions by get the transcripts sequence from the RNA-seq data. Here I recommended the tissue sample requirements for the each part. And for most projects, if you don't want to do the DNA extraction in your site, you can just send us the enough tissue size for each part. Then we will do the extraction and experiment and assembly and one-stop solutions for you in our lab. And if you want to do the DNA extraction by your side, then we recommend that you do the extraction of high quality, high molecular weight DNAs. Then this DNA quality is very important for the PacBio sequencing results. And for the RNA samples one, we have a recommendation is that the samples you prepared is better to mix from different tissues or different states or periods of the individuals that we can get more information of some transcripts that has specific expression time or positions. Then yeah, this is this part. And also for the samples for the different stems, we recommend to obtain from the same individuals to ensure that the genetic informations are same with the samples. Then we will cause less data wasting when we do the each part. For example, if you just send NGS with one sample and the TGS with one sample, then we do the back evaluation. We will find some rates that will not be mapped to the assembly based on the TGS data because some sequence of the different genome parts will be unmapped. Okay. Since we know that high molecular weight DNA extraction is very important, and for long-read sequencing, if you start with the high molecular weight genomic DNA, it will result in a higher performance and longer libraries during the sequencing. For the high molecular weight, we usually say that if the length is more than 50 kb, then we will say it's a high molecular weight. And it's enough for the sequencing read lines for the hi-fi sequencing. And here I should list some summaries for best practice for DNA extraction for PacBio hi-fi sequencing. Like you should use the fresh or flash frozen tissues, and this flash frozen tissue should be stored at 80 degrees centigrade minus and avoid the freeze and thaw cycles. And also for the blood samples, you'd better not store them longer than 2 days at 2 or 7 centigrades. If longer storage needed, should frozen and adequate. And for some others recommendations during the experiment, like the remove all the RNA with RNase A and avoid some substance that affect the DNA extraction. And also you can purchase some related kit to do this. And also, yeah, after the extraction, you'd better to have a test on the DNA quality, like the purification and the concentration and the total amount, whether it could reach the requirements of hi-fi sequencing. Yeah, also for here to refer to. And if you want to have some recommendations of the extraction methods, from our experience, we would recommend this method for you to different species. Like for animals, we recommend using the SDS extraction method with the purification kit. And also plant, we more recommend the CTAB method. For blood, we recommend this specific whole blood kit, and for cells, we recommend just the purifications. And also here, I want to share you is that there is a website that they share the publications that already has the high molecular weight DNA extraction method in these publications, and they can be divided into different class, like the animals, plants. Under each class will has very detailed classifications. Then you can find whether your target species has the information in this one. Then you can find the related information for reference. Last, I want to share our assembly experience. Since we have already get about more 118 successful cases that have been published in high impact factor journals. And we have solved many difficult species assemblies, like for the T2T, the haplotype resolved, and the big genome, and also the polyploid species. We all have successful cases for this species. And here I show one for each, and for you to refer to. Hope to give you some informations to help your start of your project. And also we have many successful cases has been published in high quality journals like the Nature Genetics. As I know, we have got 25 journals published in Nature Genetics. So we have really extensive experience. And that's what I want to talk here today. And if you have some projects want to do the genome assembly, or you want to know more information about the genome assembly, then you can contact us, and we will show you more details or information you're wanted. Okay, that's all. Thank you.